ready for Liz?
Hi.
Hey, Liz. So
Hi.
this is Liz. Liz, we first met at the University of Cardiff again. Who brought amazing gadgets that my husband tried to steal. You're amazing. You're informative, and happy birthday to your daughter who is
Thank you.
10
It's
today.
tomorrow, so yeah, apologies.
And obviously COVID has really messed that up. But
Yeah.
thank you for giving your time to us, and I'm absolutely convinced you will be fascinating with all the information you've got to share. I'll turn my video off.
Thank you. Well, it's great to see you. I'm really sorry it can't be in person, and I'm also sorry that I have to come and then go immediately after. As Sophie mentioned, it's my daughter's birthday tomorrow morning, and so I'm going to be woken up really early. So I need to get an early night tonight. I'm going to try and share my screen so that hopefully you can see. So hopefully, can you see my slides? Excellent.
Yeah.
Good. Fantastic. Great. So yeah, it's a pleasure to kind of be with you virtually. As Sophie said, we met in Cardiff, I guess earlier, about a year ago. I completely lost the thread of time at this point. And it was fantastic to meet all the UK-based families. I apologise in that they will have heard quite a bit of what I'm planning to say already, but hopefully it will be, there's some new stuff in there, and it will be hopefully of interest to you more widely. So this is a picture of my lab taken pre-lockdown, obviously. I have a couple of extra members who've joined, sadly, one of whom has never actually met the lab in person, but hopefully we'll be sorting that out fairly soon. So it was interesting to hear, I caught the tail end of the discussion prior to my talk, because I am one of those researchers who's become interested in the gene CACNA1C because of its role in other conditions, particularly psychiatry. I'm based in the Department of Psychiatry in Oxford, and my research is trying to understand how individual genes affect risk for psychiatric conditions. However, obviously working on CACNA1C, the data that I've been collecting, I think, have relevance to Timothy syndrome as well. And so I'm going to tell you a little bit about what we found out in the last few years, and hopefully flag up some areas that might be of interest to you guys. So I guess this paper here on the left is incredibly famous. So this was the publication of the human genome in 2000, and this was on the front cover of Nature and highly celebrated. And the human genome is now a kind of resource that anybody can access. So you can go to the human genome browser and look at the data for yourself. It's all there. But I think many people thought that once we sequenced the human genome, we'd understand everything. You know, we'd know what all of these genes were doing and how they might be relevant to us as human beings, and how alterations in these genes might affect risk for illness. But I think probably realistically what we've learned is how much we don't know. And so what I'm going to be talking to you about today is the process that I've gone over the last 5 years or so in illuminating exactly how much we don't know about human CACNA1C. So I don't know if you've had presentations already. This is just to remind you that the CACNA1C gene encodes the CAV1.2 calcium channel, and this is the channel that contains the mutations in Timothy syndrome. So this is kind of a cartoon of what the protein might look like if you unfolded it. It contains these 4 different kind of domains that wrap around to make a pore through which calcium can enter the cell when the cell is electrically active. And obviously I'm interested in brain cells, which communicate using electricity, but many tissues do this, including, obviously, importantly, heart. It's a very electrically active tissue. I've been working with an artist over the last few years, and she drew this picture of the calcium channel. And I love this because I hadn't really told her very much about the calcium channel when she produced this. And what you can see is that she's represented it as a square, and then she's got the little calcium ions in the center of it. And I love this because that's what the calcium channel looks like. It is a square. These 4 domains wrap round, and they kind of fold together to make this square pore through which the calcium can kind of enter the cell. So I've said that the CACNA1C gene encodes the calcium channel, But actually, one of the things that's been really clear throughout our understanding of human genetics after the publication of the human genome is that this idea that one gene makes one protein is completely wrong. We know that actually the majority of genes make lots of different, slightly different proteins from a single gene. And the way that they do this is via a process called splicing. So what you're looking at here, can you see my pointer? I don't know if you can or not. I think there is a way of making a laser pointer. Let me see if I can do that. Can you see this?
Yes, we can see it.
Oh, good. Okay. So genes are not, when on the chromosomes, they're not all in a line. Well, they sort of are, but the genes are comprised of different chunks, split into different kind of little pieces that are sewn together to make the final RNA, which makes the protein. And so in this cartoon here, you can see that there's 5 different exons, and that's what these chunks are called that are sewn together. And then there's these bits of sequence in between which get cut out. So when you want to use this information that's in the genome, the first thing that happens is that the DNA is encoded into something called RNA, which is an intermediate molecule. It's like a kind of a recipe from the human genome, if you like. And at first, this RNA contains all of the sequence. So it contains this stuff here that's eventually going to get cut out. But then you have this process called splicing, which removes these non-coding bits, these bits here that are between the exons. And this process is controlled in such a way that a single RNA can potentially give rise to lots of different proteins via this process of alternative splicing. So you can see here, this early RNA is converted into 3 different mature RNAs, which have slightly different composition in terms of the exons that are in them. So this one here contains all 5 exons, but in this one here, exon 3 has been cut out as well as these bits here. And in this one, exon 4 has been cut out. And this in turn encodes different proteins. So in this case, this is the protein that you would get if you encoded the full thing. But here you can see that by cutting out exon 3, we've lost this bit here in this protein. So this bit gets cut out. And alternatively, by cutting out exon 4, you lose this bit here. And so in this way, a single gene can give rise to lots of different proteins that are similar, but not all the same. And this is definitely the case in terms of CATNA1C. CATNA1C is a really big gene and it's very complicated. So if you go to the human genome browser at the moment, this is what it will tell you is made from the CATNA1C gene. Each of these lines is a different RNA. We call these isoforms. And you can see that there are some parts that are common to all of them. So here, the upright lines are the exons, so these bits that remain, and these long horizontal lines in between are the introns, the bits that get cut out. So some of these exons are present in all of the isoforms. You can see this one here, for example, but some of them are only present in some of them. So this little bit here only seems to be present in this isoform. However, although if you go to the genome, it's all very confidently annotated, these are the isoforms. Actually, all of this is based, for the most part, on computers' best guesses as to what the genes are making. For technical reasons, historically, people would, rather than sequencing whole isoforms, would sequence little bits and pieces, and then they would use computational approaches to try and put this information back together and figure out what the whole long isoform looks like. But the problem is, if you've got a gene like CATNA1C, it's really difficult to know what up this end is stuck to what down this end. And so this was kind of a best guess, but it's far from perfect. So the question that we've been asking is 2 things. Firstly, do these isoforms that you can see here, do these actually exist in nature? Are they real, or are they just false positives? And then alternatively, are there other isoforms that we don't know about at the moment? And how many of these are present in human brain? And I was interested, as I say, because I come from a background in psychiatry, in understanding what kinds of calcium channels are present in the human brain. So in order to do this, we had to develop a new technical approach. As I've said, historically, people would sequence little bits and pieces of the calcium channel and then do their best guess jigsaw puzzle, putting it all together. But what we wanted to do was to sequence full-length isoforms, so see what is actually present. So in order to do this, we had to have post-mortem brain tissue, which we acquired via a collaboration with a lab in the States led by Danny Weinberger. And it's really important to have brain tissue that's being collected quickly after death, because after death, RNA starts to get cut up. And so we needed the full-length RNAs to be present. And so Danny's been fantastic in sourcing really high-quality brain tissue for us. We then used a molecular technique that allows us to amplify the signal from CACNA1C. Obviously, CACNA1C is one of 20,000 genes in the human genome. So given that we were just interested in CACNA1C, we wanted to have a way of ensuring that we were just looking at that transcript. And this was work done by my postdocs, Aitani, and then Nicola Hall, who's still working with me now. What we did then was to use a new technical approach, and this was the piece of equipment that I took and showed Sophie and everyone in Cardiff. It's something that's completely miraculous to me. It's a piece of technology called Oxford Nanopore technology sequencing, and it allows you to sequence full-length RNA. So historically, sequencing, as I say, was done in small chunks. You would get maybe 400 letters of the genome out of a single piece of sequencing. But this gadget will essentially allow you to sequence as much DNA as you give it. So it will just sequence a whole thing. And the thing that's really miraculous about it is that I took it on the train to Cardiff, because it's about the size of a stapler. It's really tiny and it's very cheap. So it makes looking at RNA much cheaper and easier and more accessible. So this technique allowed us to sequence lots and lots of different calcium channels from the human brain. And then working with my collaborators at the Earlham Institute in Norwich, and particularly Wilfred Harty, he developed a computational program to allow us to make sense of all of this information. When we run these studies, we get millions and millions of what's called reads, so individual isoforms. And obviously, you couldn't really go through that information one by one. You have to have computational means of making sense of it all. So Wilfred's written analysis pipelines that allow us to do that. So what did we find? Well, as I showed you originally, what's currently on the human genome says that there's 40 different types of CACNA1C encoded from the gene. But actually, we found a lot more than that. We found 251 different types of CACNA1C present in human brain, so different isoforms. And it's not the case that the 40 that were there were correct. Actually, we only found 10 of the ones that had been previously there. The vast, vast majority of the isoforms are novel. In addition, it wasn't just that, obviously, there's lots of different exons of CACNA1C, these little pieces that can be patchworked together in different ways. You can imagine that if you had 50 different exons that you could sew together in different ways, that you could get a lot of combinations just using the known exons. But actually, we found that there are lots of chunks of CACNA1C that actually encode new sequence that hadn't been described previously. So there were 50 annotated previously, and we found another 38. Now, interestingly, within the human brain, we found that different types of calcium channels are present in different parts of the brain. And that makes a lot of sense because obviously the different parts of the brain do different things. So that says to me that these calcium channels are subtly different in terms of the way they're functioning. And that helps you to get the specificity that you need to have in different brain regions. Interestingly, we've also done some studies in mice, and I'm going to show you some of the data from that in a minute. But we found actually there are really big differences between mice and humans. And I think that emphasizes how important it is for us to study human cells and human tissues. And I know that a number of people have been involved in providing skin cells from them and their family members, which we can turn into neurons. And that's hugely helpful to us as scientists to have access to actual human cells. And then finally, I think one of the things that's possibly going to be most interesting to you guys is that as well as being differences between different brain regions, there's also big differences between the brain and other tissues. And I'll show you some of this data to explain what I mean by this. So I'm just going to start by giving you a flavour of what we found in mouse. And this was work done by my student Saeed. He's looking very puzzled here because he's just discovered that we have a vegetable steamer in the lab, which we actually use for certain experiments. And he had no idea why we had this there. So he took a selfie being very puzzled of this. So what Saeed did was to we were interested in seeing, were there differences in the types of calcium channels present in the different tissues? And obviously it's much easier to do that in mice than it is in humans. We can access heart and brain and aorta and other tissues much more easily. So what we did was to take some mice and dissect out different parts of the mouse, so brain versus heart and aorta, which is the one of the blood vessels that flows from the heart. And we looked to see what types of calcium channels, Cacno1C calcium channels, are present in these different tissues. Now, this is a kind of summary of that data. I'm not going to go through the details of what this plot is exactly, but what it allows us to do is to see how similar or different particular tissues are in terms of the types of calcium channels that are present. And what you can see really nicely is that the brain, which is shown here in green and this kind of turquoise color, is totally separate from the heart and the aorta samples. So there are profound differences in the types of calcium channel that are present in the different tissues. So this is all well and good, but this is mouse, obviously. And as I've said, what we're really interested in is understanding what's going on in humans. So what we've been able to do is to access data that have been collected by a large consortium that have collected sequencing data from different tissues of humans. And that's allowed us to take the information that we've obtained and figure out which bits of the calcium channels show differences across different tissues. And consistent with what we found in mice, we find really big differences in the calcium, the different parts of the calcium channel between different tissues. And I think this is really important for Timothy syndrome for reasons I'll come on to in a second. So what you're looking at here, the kind of lightning bolts are new stuff that we found, so new sequence, new exons, bits that are included or deleted. And anything that's in blue is a part of the channel that shows differences between the heart and the brain. We're in the process of running other tissues at the moment. We're particularly interested in smooth muscle, so colon particularly. And I was having a chat with somebody on Facebook the other day about, the fact that they've been seeing kind of gastric changes in their child with Timothy syndrome. And I think that the function of the channels in these other areas is really important. So if we look at this data here, my prediction would be that depending on where the mutation that you have in the calcium channel is, that could help to determine what kind of symptoms you see. Because if you have a mutation in a part of the channel that's more expressed in brain, say, you would expect to see changes bigger in the brain than in the heart. Whereas conversely, if you had changes that were affecting the gut, perhaps you could see bigger changes there than in the brain. And so I just want to finish by giving you an example of where Cacno1C actually is. So this is data taken from this large study that I've talked about. It's a project called the GTEx project. And all you're looking at here is the total amount of Cacno1C that's present in different tissues. And you can see, unsurprisingly, there's lots of Cacno1C present in the artery and a decent amount present in brain. But there's also really large amounts present in the colon, for example. And so I think by trying to understand what Cacno1C is present in these different tissue types, that might help us to understand, firstly, which tissues might be more or less affected by a particular mutation, but also help us to understand how this very complex and kind of multi-symptom condition comes about. So that's all I wanted to say. I'm just going to kind of summarize and then tell you a little about what we're up to at the moment. But I hope I've left plenty of time for questions, since I'm afraid, like I say, I have to disappear before too long and get to bed for my early morning start tomorrow. So I think the bottom line, if we've learned anything, it's we've learned how much we don't know. There is a vast amount that we still need to understand about Cacno1C in humans, particularly in human tissues. We've seen that it is expressed in lots of different tissues, which I think would probably be unsurprising to you guys who are seeing this day to day in your children. And what I've found recently, and this is something that we're continuing to work on, is that different types of Cacno1C are present in different tissues. And I think, as I say, that by understanding this could help us to understand why different people have different types of symptoms. So what we're up to at the moment, we're looking at Cacno1C in more tissue types than we've looked at so far. I've mentioned that we're in the very late stages of looking particularly in the colon to see how that changes, how that is different to heart and brain. I'm also in the early stages of doing a study where we're going to look and see whether the Timothy syndrome mutations affect the types of Cacno1C that are made. So normally when we think about mutations, we think about them directly affecting the protein sequence of the channel and therefore affecting the function of the channel. But it's a bit of an unknown question as to whether the Timothy syndrome mutations might also affect this process of splicing. In other conditions, it's often the case that mutations that affect the protein coding sequence can also have effects on splicing. So hopefully, touch wood, depending on COVID over the next year or so, I should be able to tell you a little bit more about that in the coming year. And then finally, I'd be really interested, once we have this information, in understanding how these differences between tissues particularly relate to the different symptoms of a given individual. So I'm going to stop there, but I'm really happy to take questions, and I hope that made some kind of sense. I apologize for not being with you in person. Hopefully next year we will be able to meet in person, but I'm, like I say, very happy to take questions. And I'll stop sharing so that I can actually see you, because at the moment I'm talking to a void.
I think that slide, Mary, and I think that slide, the second to the last slide that you had up there with all the breakdown.
Yeah.
Oh, sure. I can pick that up. Hang on.
Yes. Could you?
That was an amazing slide.
Yes.
So
That's going to help with my screening arguments with
this one.
Yes.
Whoa.
I can send you this picture.
Yeah.
I can make it a bit prettier. But yeah, I think this is crucial, because people think about Timothy syndrome, they think about the heart and they think about the brain, but there's all these other tissues that have some level of CACNA1C expression. And I mean, I'd be really interested to know from your experience, from Catherine's experience, you know, any tissue that's electrically active, muscle, you know, across the body is electrically active, seems to have some kind of CACNA1C expression.
That is like gold.
I think it's so interesting because I'm seeing bladder on there. I don't
Yep.
see kidney though, and
Kidneys here, so there's not much in kidney, not huge amounts. But yeah, I mean, the other thing to bear in mind is that these are quite crude studies. The way this study has been done is they've dissected out quite large chunks of tissue and then just kind of smushed up everything together. So you don't get any information about what's going on at the single cell level. And so it's possible that there's a small number of cells that might, within a tissue, that might have much more CACNA1C. But I hope this is useful. I thought you might like it, which is why I put it here.
Why is the brain split into so many bits?
They're just different parts of the brain. So broadly speaking, there's different parts of the cortex. So the cortex is the bit, when you look at the outside of the brain, it's the bit you see. It's the kind of really folded bit. And different parts of the cortex do different things. So the frontal cortex does a lot of the really complicated stuff, so decision-making, planning, that sort of thing. And then parts at the back of the brain, the cortex at the back of the brain is involved in visual processing and processing sound information. So they've separated out these different parts of the brain because they do different things. And then other parts, the cerebellum, for example, is really important for coordinating movement. Sorry, Sophie, go for it.
Where's speech? Where's speech?
They haven't really got speech on here. So that would be, it's the temporal cortex. I mean, the hippocampus is close, but it does something different. The hippocampus is about memory. So Jeremy's done quite a lot of work. Jeremy Hall has done quite a lot of work on understanding what CACNA1C does in the hippocampus. It seems to be quite important there, although there's not vast amounts of it. But they haven't got the temporal cortex on here, unfortunately. Yeah. But I suppose, broadly speaking, when we did our study, so we have looked at other brain regions that aren't on here, and we found typically most of the cortical regions looked fairly similar. And I think that's probably to do with the types of cells that are present in different types of the brain. So most of the cortex contains a particular mixture of different types of brain cells. And then the brain cells that are present in the cerebellum are very different. So when I said we saw differences across brain regions, the cerebellum looked profoundly different from all the cortex regions.
Wow. God,
I'm really happy to share these slides with you if it's helpful.
Yes, please. Yeah.
this is amazing.
Oh, sorry.
Wow. So I know other people might have questions, but which one of these to you is of concern that's not being addressed at the minute?
I think that's a really good question. I mean, to be honest, I'd be guided by you guys. What symptoms do you see that you don't think is really appreciated clinically at the moment?
definitely speech,
Yeah.
Yeah.
just from a personal point of view.
Where would the immune system fall? on
Yeah.
that?
There's not huge amounts of immune stuff in this example. So there is whole blood somewhere. Oh yeah, that's useless. There is a little bit of CACNA1C in certain blood cells, but it's a particular population of blood cells that seems to have CACNA1C, and it's very low overall. They haven't got any of the sort of classic immune tissues in this study. But it's interesting that you flagged that up. I mean,
there are some lymphocytes, although they're transformed.
they're transformed, yeah. So
But I also want to mention that, you know, in our experience, and I think in many other groups' experience, the levels are not necessarily always the most important. I mean, you can have very low levels of CACNA1C or CAV1.2, because that's how we call the protein, and that can have a dramatic effect on a cell.
yeah,
So
absolutely.
I also want to mention this because even across brain regions, actually, you know, CACNA1C is one of the most highly expressed channels. I mean, there are a lot of channels. It's one of most, not highly, but broadly expressed. Many other channels have not the same, you know, I guess, you know, I think broad expression that we see because they're so essential to the function on every single neuron. Right? Alpha calcium channels are absolutely essential for translating electrical signals into biochemical systems inside the cells. So in a way, I don't know whether we would expect even across the brain to see like major differences. I don't know whether, Liz, you
We
would
see differences in splicing profile, so that the kinds of the precise details of the splice variants differs quite a bit. I mean, but it's
the splice, yes,
relative difference. It's not that we have some channels that are only present in some regions and some that are only present in the other.
Right.
So far.
Yeah.
But it's more that different brain regions have different sort of relative abundance of particular variants, which I think that's about the types of cells that play,
exactly.
right? Different cells need different types of calcium channels.
I think that gives like different nuances to the type of activity of these channels. But
yeah,
we've actually done a lot of other experiments, like even molecular biology, and we find that alpha calcium channels in general are some of the most stable in terms of expression. You know, it's very difficult actually to interfere with this. And I don't know how you feel, Liz, about the studies that have been looking at gene expression across even individuals in the general population in the brain with some of the common variants. I you know, it's always been surprising to me that these levels are actually rather not across individuals and across brain regions quite stable.
Yeah, I think that's fair. I mean, I guess, you know, what we haven't done in huge detail yet is to look at that systematically. So now that we have information about what the full length channels look like, we can go back to data sets like this one. So this is an example of a data set where it's little pieces of sequencing that you have to sew together. Now, in order to sew that together accurately, I guess it would be like, you know, it's much easier to do a jigsaw if you know what the picture is. If you have no idea what the jigsaw is meant to be in the first place, you're really going to struggle to put those pieces together. So what we're doing at the moment, working with Wilfred particularly, is saying, okay, so now we know what the full length channels look like. Let's take this short sequencing information and see if we can sew it together better than we could before and look at a better level of detail, essentially.
I'm a little bit interested in what this doctor, I can't see him on the screen now, but said about the lymphocytes, because we just got, as I said earlier tonight, we just got some results from my daughter's colon, which was sent to England, and then we got back like an increase of lymphocytes in plexus mesentericus, and it says it's like 3 to 4 T lymphocytes around ganglions and infiltrated in ganglions, which is quite.
That's probably sort of, that might be kind of, I guess, infection related or immune related. So most of the T and B lymphocytes, which are these different cell types, do different things in the immune system. So they're white blood cells. So obviously the red blood cells oxygenate you essentially and keep, you know, move the oxygen around your body. But the white blood cells are really important for regulating infection. So looking out for invaders essentially and getting rid of them. And so it may be that some of those changes are related to infection.
Yeah, because it says there it's quite high, the colon sigmoid thing on the screen, and she just had to remove her sigmoid, like 30 centimeters of her colon. the sigmoid, and then she got an colostomy.
Wow.
So it's
Wow.
quite fascinating to see this on the screen.
Yeah, I mean, I was utterly unsurprised when I heard that people were having kind of colon issues of various sorts, because it's a contractile tissue, it's electrical, and it has a lot of CACNA1C in, has a lot of CAV1.2 in, so it's not at all surprising that you would see changes. Hi, sorry, I've just seen Marianne's visitor.
Liz, why is uterus so high? What's going on there?
Again, it's contractile. Anything that's needs to contract. That's what the uterus does, right? It expels babies. So I mean, I think most muscles will have a decent amount of CACNA1C just because they're electrically active, and so in the brain, the calcium channels in the brain do slightly different things in different tissues. So in the heart, it's really important for kind of coordinating the heartbeat. In other tissues, it's important for sort of ensuring that signaling sort of happens at the right time. And in the brain, it kind of coordinates between short-term changes in activity and long-term changes in what the cell is doing. It regulates the expression of other genes, essentially. So it's a way of linking the electrical activity of the cell with what the genes in that cell are doing. Does that sort of make sense?
Yeah.
So would they have problems giving birth, or a risk of women with Timothy, if they were to have children, would they struggle to have contractions, or would they give birth prematurely, or need?
I have no idea. I mean, I have no idea. I mean, you'd predict that something would be, but I guess it depends a bit on, as Sergio was saying, the channels are doing different things. It's quite difficult to perturb their function often. You may well see that in some of these tissues, actually, changes in this particular channel aren't a big deal, because the body sort of compensates for it by changing the activity of very similar channels. So this is one of a whole suite of related channels. And often, in the brain, we see this a lot. If you mess with one thing, you often get compensatory changes in other things to kind of even the balance out. It's the body's way of trying to compensate for something not being quite usual.
Just to give you a sense also clinically, blockers of L-type calcium channels are used sometimes in the clinic to prevent premature birth.
Oh, interesting. I didn't know that.
Or some of the blockers are used because the uterus is trying to, when you have premature contractions, one of the ways in which you can buy some time is by blocking precisely these channels. And so there is evidence that they play an important role.
That's interesting.
Contractions,
I didn't know that. I mean, we've been mining kind of health records, because obviously lots of people are prescribed calcium channel blockers for angina and high blood pressure. They're really widely used. And there's been this sort of history of people maybe thinking they might be useful for treating bipolar disorder, particularly, which is my primary interest. But we looked at the evidence, and it was really sketchy, in that the evidence just wasn't good enough to conclude anything. But now, as we have better digital records, we're able to mine that information and see whether people that are given calcium channel blockers because they happen to have high blood pressure have a reduction in the chance of getting a psychiatric illness. And it looks like they probably do. It's quite difficult data to interpret, obviously, because we haven't chosen who gets calcium channel and who blocker and who doesn't. But certainly in our hands, at least in the couple of data sets we've looked, it does look like there's some indication that blocking CACNA1C and related calcium channels with these blockers might be beneficial in terms of psychiatric illness.
Liz, I don't know whether you were on earlier, but probably not. But we had a small discussion about there's quite a few of our kids that are eating and eating without knowing when to stop.
Oh, okay.
Which was not really looked at. We've not really discussed, as far as I know. But where would that fall on this chart? If Calvin
interesting question.
eats and eats and eats and eats and eats, and you almost have to. He will eat 2 adult-sized portions for dinner, and he will just keep going. But his metabolism's where would it go?
That's complicated. So control of what we would call satiety, knowing when you've had enough to eat, is really complex. So it involves the gut, but it also involves the brain. And there's quite a lot of communication between the gut and the brain. They talk to each other quite a lot, as you might expect, because knowing how you're doing nutritionally is really important to survival and your function. So your stomach is always saying to your brain, hey, I've got enough glucose and not enough of this. And, you know, there's a lot of back and forth. So it's possible that some of it could be happening in the stomach, but also there's a large kind of systems in the brain that figure out how full you are or how hungry you are. And there's some really very rare conditions that are due to a mutation in a particular receptor that essentially turn off the ability for the brain to know when it's full. And so kids that have this condition are permanently hungry. And so the bottom line is, I don't know, it could be brain, it could be peripheral, it could be a bit of both. It's kind of an interesting question, that.
If it was going to be brain, where would it be? Or do you don't know?
It's so the brain is kind of composed of, I guess, 3 different levels. So you have the kind of core of brain is what we call the brain stem. And this does the stuff that you need to keep alive. So it controls your heartbeat, it controls breathing. So even very primitive species have a brain stem. On top of that, you have another level of control, which is we call the kind of subcortical regions, that tells you another level of information you need to stay alive. So it tells you to forage for food, and it controls finding a mate and that sort of thing. And then on top of that, you have the cortex, which is this sort of bumpy bit that we've already talked about. And that controls these other regions. So that's the reason, essentially, that as humans and primates, we have a level of self-control, because the cortex tells, you know, the brainstem to be quiet for now because I'm doing something else. So most of the kind of appetite stuff happens outside of the cortex. However, I can see it's also very possible. So my father had a stroke 20 years ago. He lost a lot of inhibitory control, and he also eats vastly more now than he did before his stroke. Obviously, this is not in any way kind of a controlled experiment, but I think a lot of his increased appetite is actually due to his inability to say, maybe I shouldn't eat that large chunk of butter. Maybe that wouldn't be the best thing for me in the long term, you know, because his brain at the moment is going, yep, I want the butter, give me the butter. So again, it's even within the brain, it's not entirely straightforward to know where that might be happening. It might be that the basic mechanism that says, you know, your stomach is empty is which would be kind of brainstem, this very kind of primitive brain, that could be where this is happening. But it also could be at the level of the cortex telling those parts of the brain to kind of quieten down for now because we're doing something else. Do you see what I mean?
Yeah. I think it's a quieten down thing. Possibly that's where the toileting issues come from, where they get carried away and forget that you're doing something and it's going to the toilet.
Yeah, they don't want to break away.
Yeah.
That would make a lot of sense.
Could I ask if anything of this is like visible on an MRI, like the brain stuff?
Probably not really. I mean, you might see it wouldn't, not on a clinical MRI. They're really designed to detect kind of structural changes in the brain. So, you know, like fairly gross, like this bit's much bigger or this bit is much smaller. These changes are much more subtle. You know, the tissue will still be there, but it's just acting in a slightly different way. It's possible you might see it in a research type scan, because we have different types of scans that we use for research that allow us to try and understand what different brain regions are actually doing functionally. Now, I don't know, I mean, Catherine would know, has anyone done sort of functional imaging studies in Timothy syndrome, or?
We just did a normal MRI and the normal MRI came out fine.
Yeah, it probably would do. I mean, that, like I say, they're really designed to detect big hole, you know, they're designed to detect, you know, damage, really quite large damage. So you see things like a stroke, you see things like, you know, advanced Alzheimer's, but you don't see subtle changes at all.
Yeah, because
to my knowledge, excuse me.
Sorry.
The doctors don't acknowledge, you know, the difficulties that comes from the brain if they can't see it. And since syndrome is so rare.
Excuse me, but we haven't really had any clinicians really look at Timothy syndrome yet. It's all been in the research arena. And so none of the children have really ever been evaluated for anything with their MRIs or anything. But Liz, this has been wonderful. And I know that these moms would love to pick your brain forever. But but we probably should move on. But I would like to suggest that possibly in the future, maybe Sophie could have could arrange for a Zoom for all the moms that might be interested.
Yeah,
And
absolutely.
maybe you would be willing to answer many, many questions that these mothers have. I was wondering why on your little chart you didn't list any of the non-excitable cells that we know that are affected by the calcium channel. So
I mean, this chart. Oh, anyway, we can come back to this. I don't want to cut somebody else's time.
I think maybe we should focus on the brain for the June family weekend. Maybe we should focus on. I mean, we've got you. We've got the University of Cardiff. We've got the whole team.
Yeah, that'd be great.
Thank you very much for your time, Liz.
I'm so sorry to have to dash. Like I say, I really need to get to bed because I'm going to be woken up at
totally
6 o'clock tomorrow.
I hope your daughter has a lovely birthday tomorrow.